In the previous video, we have understood how this IRF7 modulates the lipid metabolism on the pancreatic cancer cells. In this video or in this figure, we will understand how this IRF7 modulates the lipid metabolism of the pancreatic cancer cells. That is, as the heading suggests, that it will modulate the RPS18 transfer from M1 macrophage to the PDAC. This RPS18 helps in this pancreatic cancer cells lipid metabolisms or pancreatic cancer cell survivals. So from the literature they have found out that IRF7 usually target the promoter regions of this RPS18, and from their analysis they are able to find out the binding regions, that binding region of this RPS18 promoter from where this to where this RPS17 gene targets and disrupts this RPS18 transfection. So once they have identified their first they identified their binding region of the promoter, what they did is they check this binding regions presence within the binding regions presence they check this binding region presence within their macrophages, and they can see that RPS18 contains RPS18 contains this kind of binding regions, whereas for this control IgG, it does not contain these binding regions. It does not contain the binding region, and this is the input. And from there, they have separated out this IgG and RPS18. So after this, they want to check this impact of this RPS18, impact of this this IRF7, impact of this IRF7 on this RPS18 formation. So what they did is they overexpress this IRF18 cells through the lentivirus transaction, and on the other side they take the control cells. And in the wild type, where they did not mutated this binding region, they can see that there is a decrease in the RPS18 formations or the luciferase signals, as this RPS18 is also co-transfected. So let me first define how the experiment is done. So in this panel C, what they did is they co-transfect this IRF7, and also they co-transfect with this fluorescence reporter combined with the binding region of this RPS18 promoter. So, and the second plasmid is this IRF7. So when they in the wild type cells, where that is where they do not use the mutated binding regions of the RPS18, they see the decrease in this RPS luciferase signals or the RPS18 signals when the IRF7 is overexpressed. However, when the binding regions are mutated, they do not see any difference. So indicating that is the binding regions that cause the RPS18's translation disruptions, binding regions of the promoter. So after that, they also check this using the DIL, where they can see that in the pancreatic cancer cells, the amount of exosomes which are red in color, and also in the BXP3 cells, the amount of exosomes which are red in color are still shows. Here they use the exosomes that are separated out when that are separated out from the M1 macrophages, wild type M1 macrophages. So in the PBS, where there are no cells, they can see the decrease in the signals because exosomes degrade. However, in the PANC1 and BXP3 cells, they can see that exosomes are still there, indicating that the exosomes are internalized by this PANC1 and PC3 cells. Thereafter, they check this IRF7 and RPS18 in the also in the case of this knockout cells and the wild type cells. So in the IRF7 knockout cells, since IRF7 is knockout, they see that there is a high amount of this there is a high amount of this IRF7 in the wild type cells compared to the knockout. However, for this RPS18, they can see that for the RPS18, they can see that the RPS18's formations are higher in when the IRF7 is knockout. So this also substantiate their results that since now IRF7 does not bind to the promoter regions of this RPS18, it does not interrupt the RPS18 transfection. And they also validated their results using Western blot, where they can see that in the IRF7 knockout there is a more presence of the RPS18 compared to the IRF7 wild type, and this Western blot has been quantified on the right hand side, where we can see that similar result where RPS in the RPS18s are shown are significantly higher, statistically significantly higher in the case of IRF7 knockout. And thereafter they check the IRF7 and RPS18 in the exosomes, and this panel F represents the results for the M1 macrophage, whereas panel G represents the results for the exosomes, M1 exosomes. Here they do not see any changes in the IRF7. However, they see the changes in the RPS18, indicating that when the IRF7 is knocked out, the exosome carries more RPS18, and that can be translated to the pancreatic cancer cells. And when they check the relative expression of this IRF7 in the pancreatic cancer cells, they find out that IRF7 does not have any impact. Does not have any impact of the macrophages IRF7, whether it is knocked out or it is present on the pancreatic cancer cells IRF7, as its expressions levels are statistically non-significant, both in the case of pancreatic cancer cells and BXP3 cells, with with macrophages containing IRF7 and without macrophaging and uh, with macrophage not containing IRF7. And when they check the RPS18 content in the RPS18 content in this exosomes, and they can see that in the case of PANC1 cells, the exosomes contents are the not exosomes, the RPS18 contents are higher in the case of this IRF7 knockout, whereas when the IRF7 is present, they can see that reduced amount of this RPS18 expression. So indicating that it's the RPS, it's the RPS IRF7 that interacts with this RPS18, and also this if there is no RPS IRF7, the RPS18 can be translated uncontrolled in an uncontrolled manner and be transferred via exosomes into the pancreatic cancer cells, as the pancreatic cancer cells also has high amount of RPS18s when the IRF7 is knocked out. And they verified their results also with the Western blot, where they can see that in the case of knockout, the pancreatic cancer cell has more RPS18, and also BXP3C cells also has more RPS18, and the quantifications are on the right hand side, where we can see that PANC1 cells has more of this RPS18 expressions compared to the wild type case, and same results are observed for the BXP3 cells. And thereafter they use the inhibitors, that is the GW4869. This is the inhibitors to prevent extracellular vesicles translocation. So when they use these inhibitors, they can see the reduced translocations. They can see that RPS7 transfer from the M1 macrophages into the pancreatic cancer cells reduced, indicating that it's the extracellular vesicles that is responsible for the transfer of this RPS18 from the M1 macrophages to the pancreatic cancer cells. And similar results they have observed for similar kind of results they have observed when they use the overexpression RPS18 in the macrophage using lentivirus in the M1 macrophages. There they can see that RPS18's transfer increased compared to the normal case where there is no overexpression of the RPS18, and similar results are observed for the BXB3 cells also. And after confirming from the several experiment, after getting the same results from the several experiment, they wanted to check what are the genes that are dysregulated that might play a role towards this, that might play roles in a way that this more RPS18 transfer to the pancreatic cancer cell leads to its more aggressive properties. So in this case they use two kinds of cells, one which has two kinds of cells to plot in this volcano plot. That is, one has high overexpression of this RPS18 and one does not have high expression of this RPS18. That is the cell systems from the panel L, and they can find out that 5 genes that are dysregulated. 1, 2, 3, 4, 5. 4 are downregulated and 1 are upregulated. And when they check their results, when they also simultaneously they did also they also analyze the role of RPS18 to the interaction of RPS18s to which kind of genes these RPS18s can RPS18 can this influence. And from and so before going to that, let's understand this panel N. In the panel N, it's the heat map plot of this dysregulated genes that are found in the volcano plot. And here we can see that the here we can see that this ILF3 is been upregulated. ILF3 has been downregulated compared to the case of compared to the case ILF3 has been downregulated in the wild type compared to the case of this IRF7 knockout exosomes. And however, for the other genes, it's reverse. That is, other genes are upregulated. Other genes are downregulated. Yeah. So let me repeat it. ILF3 is upregulated in the case of IRF7 knockout, and other 4 genes are downregulated. And after identifying these 5 genes, they decided to check the relationship between RPS18 genes and this dysregulated gene. So what they did is they first identify the genes that are influenced by this RPS18, and they can with the help of this biogrid, and they are able to find out 373 genes are identified with are influenced by RPS18. And when they compare it with this dysregulated genes, they can see that the ILF3 genes is the only gene that is overlapping. So they check this ILF3 expressions in the wild type exosomes, and this ILF3 expressions in the experiment that has these wild type exosomes, and macrophages that are normal macrophages or wild type macrophages released exosomes, and the IRF knockout macrophages release exosomes. And they can see that ILF3 expressions are upregulated whenever this IRF7 knocked out exosomes are used, both in the case of pancreatic cancer cells and the BXP3 cells. However, from the M1 in the exosomes, they do not see more ILF3 expressions, indicating that this RPS18, when it's transferred to the pancreatic cancer cells, then it upregulates the ILF3 expressions that might help the pancreatic cancer cells towards its aggressive behavior. So I hope you understood how they find out, or how they come to a point that IRF7 modulates RPS18 from M1 macrophage to pancreatic cancer cells, that in turn upregulates this ILF3 expression, and that could that could be the reason for the aggressive behavior of the pancreatic cancer cells, or one of the reasons. But that thing is not covered here, last part. We will see whether this point is covered in the subsequent videos or not. Till now, if you need to understand these experiments and the previous experiments, and also the future or the next experiment that we will discuss in our future videos, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.